Welcome to this presentation of our work in synthetic biology in Pheodactylum tricornitum. Pheodactylum tricornitum is a well-studied marine diatom with great potential as a sustainable growth platform for high-value compounds such as terpenoids. Terpenoids are secondary plant metabolites which are of interest as pharmaceuticals, cosmetics, and dietary additives. They often occur at very low concentrations in their native species, making isolation on an industrial scale unfeasible. Due to the high complexity of the molecules, stereosynthetic routes have low efficiency. One way around of those problems is combinatorial metabolic engineering of microbial or plant hosts. Pheodactylum is a model diatom with a fully sequenced genome, stable transformation protocols, and readily available molecular cloning parts. In our lab, we use Golden Gate cloning for the rapid assembly of constructs. Here we introduce two diterpene synthases, LPPS and TPS3, into Pheodactylum to produce the diterpene mannose oxide. The transgene expression cassette contains BLE, the zeosin resistance gene, and the diterpene synthases fused to fluorescent proteins for high-throughput screening. The two constructs differ in their codon optimization of the diterpene synthase coding sequences and intron numbers. The top construct, C10, is Pheodactylum codon optimized, whereas the bottom one, C9, is optimized for expression in the green alga Chlamydomonas reinhardtii. The C9 construct additionally has regular interspersions of the RBCS2 intron 1. Both introns and codon optimization has been shown to enhance transgene expression levels in Chlamydomonas. When growing diterpene-producing strains, it is desirable to be able to control at which stage the diterpene is generated. The endogenous promoter of the methionine synthase gene, MET-E, allows for B12 suppressible transgene expression. Both diterpene synthases are under the control of the MET-E promoter, which acts as an off switch. In the second figure, we can see that YFP fluorescence is switched off upon B12 supplementation. Having established strains that reliably produce mannose oxide, we next aim to increase mannose oxide production. We tested whether introns and codon usage impact transgene expression, similar to what had been reported in Chlamydomonas. Looking at this data, it is clear that introns and codon optimization do not affect expression levels of exogenous DNA in Pheodactylum. Lastly, we hope to achieve a higher mannose oxide titer by increasing the cellular pool of the precursor molecule, GGPP. To this end, we overexpress two enzymes involved in the GGPP biosynthesis, namely DXS and GGPP synthase. Our data shows that neither overexpression of endogenous nor foreign GGPPS enhanced the mannose oxide production. However, when DXS was overexpressed, as can be seen in blue in the graph, we found a significant increase in mannose oxide measurements. To summarize, we successfully engineered Pheodactylum to produce the diterpene mannose oxide under MET-E regulation and tested two approaches of increasing transgene expression levels.